So good morning, students. Good
Good
morning.
morning,
Morning,
sir.
sir.
So I see just only 7 students have joined. So by the time others join, do you have any questions? Any queries? Anything you want to know? Okay, so by the time others are joining, let me ask you few questions uh, related to immune response. So we yesterday started with immunology, the very basic idea about immunology. So anyone, uh, when we say that there is a first line of defense, so what are the first line of defense? Anyone? So which is the first line of defense in our body? Okay, I can see one hand. Any other person? Okay, okay. So maybe, so you just need to switch on your camera while giving the answer. So Sanjana, go ahead.
Okay, sir. The first line of defense includes the skin, which is non-specific in nature and helps us uh, and acts as a physical barrier. The next that we have is the mucus lining, and the third is the secretions. The secretions, for example, the saliva in mouth and the acid that is secreted in stomach.
Okay, okay. So yes, these are some of the first line of defense, and you know, I'm not discussing these defenses in detail at the moment. But if you read about skin, right? So there are so many factors on the skin, right? There are like sweat, there are various enzymes, there are raw antimicrobial proteins, there is a normal microflora that, so all these things they do not allow the growth of pathogenic microorganism, right? So skin is very important. It prevents us from infections. But yes, if there is some cut, if there is some problem with the skin, then you can get infection. Then, as Sanjana said, there is mucus lining, right? So what mucus lining does is it tries to like bind to the pathogen, right? And then try to uh, like release or the pathogen out, right? So that is how, and they are non-specific. Why they are non-specific? Because they don't see whether it is bacteria, it is fungi, or it's a dust particle or whatever. So they just trap and they just remove, right? Okay, and if we talk about the second line of defense, so students, second line of defense, okay, Shakti, yes,
Am I audible?
yes, Shakti, you can also switch on your camera, yes, Shakti,
Yes, sir. So they are phagocytic white blood cells and also some antimicrobial proteins, and they are also non-specific in nature.
Yes. So the second line of defense again, it includes some of the white blood cells. Okay. Uh, so as I said, there may be some chances where the skin is breached, right, and the pathogen may enter into the tissue spaces. So in that case, what happens? These phagocytic cells, they can go and capture the pathogen, okay, and they can kill the pathogen. So how these pathogen capture are being captured by these cells, and how these phagocytic cells are killing the pathogen inside them, okay. So we will be reading about them when we will be reading about the different cells of the immune system, okay. And then the third line of defense is our proper immune system, which includes the B lymphocytes and the T lymphocytes, and they are specific, right? Like an antibody which is made against a specific pathogen, it is specific for that pathogen, right? Antibody which is made for, suppose, uh, mm, dengue virus, it will not go and bind to the SARS uh, COVID virus. Okay, so these antibodies are specific, so they are termed as specific defense. Okay, okay. My another question would be, so I think you all understand that immune system is really required because it can help us in preventing diseases, right? So it fights for us and it eliminates the pathogen, right? So uh, can there be some instances where this immune response act against us? Can this happen?
Yes,
Okay,
yes,
so
sir.
okay, Sanjana, you want to answer this?
Sir, so in the case of autoimmunity,
Sanjana, you
uh,
can switch on your camera
okay, sir.
while giving answer.
So,
Yes.
sir, in the case of autoimmunity, where our immune system is unable to distinguish between self cells and non-self cells, so it attacks its own cells. And also in the case of hypersensitivity,
Yes.
and hypersensitivity example is allergies, against which Ig antibodies are produced. Sorry, IE antibodies are produced.
IgE.
Yes, sir, IgE antibodies are produced.
Yes. Yes.
That's
Okay,
it.
good, good. So we will be reading about immunology, right? So we always have this in mind that it is going to help us. Yes, it is very good for our system, but in few cases it can lead to some issues, like she said, in autoimmune cases, where one of the factor of the immune response, which is recognition of self and non-self, right? So there can be a problem, and that can lead to autoimmune disorders. And when we will read about autoimmune diseases, you will realize that there are so many autoimmune disorders, right? And they can affect any organ, right? Or they can be, they can attack multiple organs, right? And then we have allergic responses, the hypersensitivity reactions. And when we will read about hypersensitivity, I will tell you that there are four different types of hypersensitivities, right? So one of them is allergy, in which IgE is involved, as Sanjana said, that in allergies IgE is involved, but there are three more types in which other components of the immune systems are involved. Okay, so again we will talk about that in detail. I think that's in your third module, right? I think. Okay, Nehal, yes.
Sir, memory cells comes under which defense?
So they are the third line of defense because they are part of the B cells. Okay.
Okay, sir.
And again, when we talk about memory, it is also specific because uh, it keeps in mind the pathogen that it has seen, right? So it is specific against that specific pathogen. Okay. Okay. So I think the way we are discussing, things are getting clear to you. Let me see if there is any other question I can ask. Okay, okay. So what I'll do is I'll just show you now a small video of what we have learned till now. Okay, and after that we will talk about how the different cells of the immune response were discovered, how other pathways related to immune response were discovered. Okay, but let's first see a small video.
We're spending 6 episodes on a special series on vaccines. Last week, smallpox immunity. After this, it was quite a while before any new vaccines appeared on the scenes. We saw the first laboratory-developed vaccine in 1879 when Louis Pasteur weakened cholera bacteria in the laboratory for use in an immunization.
So students, you remember like uh, we always start the history of immunology with the work of Edward Jenner, right? Who developed the vaccine for smallpox, right? And then the next vaccine Louis Pasteur developed, right? So that was the vaccine which was developed in the lab, and uh, you can see he started with the work on cholera. Okay.
He accidentally discovered that this was possible when his assistant forgot to inject chickens with a fresh batch of bacteria before a holiday.
Okay. So as I said, yesterday I also talked about it. I showed you a picture also that it's a very interesting story. Accidentally, that was discovered that there can be attenuation, right? So as the video said, that uh, the person who had to inject the fresh culture into the chickens, he forgot, and later on, so the old cultures were injected into those chickens, and the chickens survived, right? So it was an accidental discovery. Let's see it again. Okay.
In the laboratory for use in an immunization. He accidentally discovered that this was possible when his assistant forgot to inject chickens with a fresh batch of bacteria before a holiday, injecting the chickens instead when he returned a month later. These chickens survived, showing only mild symptoms, and later proved to be immune to further infection. Pasteur figured out that the bacteria had been weakened by the prolonged oxygen exposure, thus creating a less deadly but still effective antigen. Other lab-based vaccine advancements followed, including the weakening of anthrax bacteria with carbolic acid, weakening of rabies virus via the drying of infected nervous system tissue from rabbits, weakening of diphtheria toxin and cholera bacteria by heat treatment, and weakening of a measles virus strain by passing it several times, like 40 to 80 times, through different cell types. These and other methods, including the use of formalin, a solution of formaldehyde, were experimented with to kill or weaken viruses, toxins, and bacteria in order to create safe and effective vaccines. In 1926, it was discovered that aluminum salts could help to increase the effectiveness of the diphtheria toxoid. We now call such substances adjuvants and make regular use of them. Might be good to stop here and explain the different types of vaccines. We'll start with live attenuated and inactivated vaccines. Live attenuated vaccines are made by weakening the disease-causing virus, meaning it contains live virus to induce immunity, but in a form that cannot cause serious illness. We should note that these types of vaccines could revert to the original disease-causing form, though this is only known to happen with the live oral polio vaccine. The polio shot, not a live virus. Inactivated viruses have been inactivated with heat or chemicals like formalin and are thus not alive and able to replicate. The response to these vaccines doesn't resemble natural infection in the way that live virus responses do, and they often require multiple doses and then boosters. Despite these limitations, inactivated viruses are sometimes necessary due to limitations of live vaccines, such as their storage temperature needs or the risk of live virus exposure in cases of a weakened immune system. There are also toxoid vaccines, which target the toxin produced by a disease-causing germ. Toxoid vaccines are used to vaccinate against diseases like diphtheria and tetanus. And lastly, there are also subunit and conjugate vaccines, which we'll get to in a bit. Production techniques have also undergone enormous changes over the years. It was discovered that viruses like rabies and polio that affect the nervous system could be cultivated in chick embryos, which helped to avoid major side effects that result from cultivating a virus with nervous system tissue. A method was also developed to cultivate poliovirus in monkey kidney cells and other viruses in dog and rabbit kidney cells and in duck embryos. These discoveries also helped to reduce our reliance on using live animals like monkeys for growing and testing viruses. However, researchers began shifting to the use of human cells around the 1960s, following the detection of simian virus in monkey kidney cells, which prompted some concerns, though later research found no relationship between the presence of that virus and vaccine complications. This does mean that many vaccines involve the use of cell lines derived from human fetal cells, which is a complex and controversial topic, one that would take an entire episode, if not series, all on its own. In 1948, the first combined vaccine became available in the United States. It combined the diphtheria, tetanus, and pertussis vaccines. In 1952, Jonas Salk began the first human trials with the killed poliovirus vaccine. The vaccine was licensed by the U.S. government when results were announced in 1955 with 80 to 90% effectiveness against paralytic polio. In 1981, we saw the first subunit viral vaccine for hepatitis B. Subunit vaccines include only the components of a pathogen that stimulate the immune system, as opposed to including the entire pathogen. These vaccines often require the use of those adjuvants I mentioned earlier since they aren't generally strong enough on their own. All HPV vaccines licensed in the U.S. are subunit vaccines, using a protein that self-assembles to form empty shells that resemble HPV virus-like proteins. And in 1986, a hepatitis B vaccine that did not use human serum became the first vaccine based on recombinant DNA technology, which describes the process of joining DNA molecules from two different species together. This sounds pretty Frankenstein and has thus sometimes been used to scare people, but it's actually pretty straightforward once you understand what the process actually entails and achieves. For this vaccine, the process involved altering yeast cells so that they produced the virus's antigen, the component of the virus that elicits an immune response, which in this case was a surface protein in the virus. This was done by using an enzyme to remove the surface protein and then insert the code for that protein into yeast cells so they could grow a bunch of it in order to manufacture the vaccines. It's an incredible feat of technology. It's also safe and effective. In 1987, a conjugate Hib vaccine, as opposed to pure polysaccharide vaccine, was licensed. Polysaccharides are long chains of sugar molecules that create an outer coating on the antigens of certain bacteria. Pure polysaccharide vaccines are able to induce antibodies against these, but aren't all that effective in children younger than 2 because their immature immune system struggles to recognize the coded antigens. These vaccines also lack a booster response upon repeated doses. These issues were resolved with the development of conjugate vaccines, which linked the polysaccharides to a carrier protein, an alteration that resulted in a stronger immune response, including a response in children. Though we should note that there are still pure polysaccharide vaccines available for a small group of diseases. We could go on and on for days about all the individual developments, advancements, trials, and people that have contributed to vaccines as we know them, but we've done our
Okay, students, so he's talking about the different types of vaccines, right? And what were the problems with those vaccines and how those problems were overcome, right? Like, just for example, the polysaccharide vaccines, they had some issues. So they were linked to a carrier protein, so those issues were resolved. Similarly, usage of adjuvants, it increased the efficacy of the vaccines, right? So we will also talk about this in because one of the module is totally dedicated to vaccines, the different types of vaccines, immunization program, and how we can grow these uh, organisms for producing vaccines. So we will obviously talk about these vaccines in detail, and I stopped here just to show you this picture, and you can see that these scientists, they are working with eggs, right? Uh, do anyone know what are they doing? Any idea what they are doing with these eggs?
No, sir.
Yes, Sam?
Sir, sir, I have two options. Either the immunity, giving vaccines to the chicken in order to develop an early immunity, which
Okay.
now I think which is not the case, or they're trying to develop, make the chicken develop some type of uh, hormone or some type of chemical which will later be extracted from them.
Okay, I appreciate the way you have thought, okay, but that is not the right answer, okay? I will tell you. Any other person who wants to make a guess like Adish did? So that was a good guess, I think. Yes, any other person?
To vaccines as we know them. But we've done our best to cover.
Okay, so I will tell you what they are doing. They are basically growing virus in these eggs, okay? So, uh, see, if you want to grow bacteria, we have already seen, we have already done the experiment. Most of the students were here in the lab and they saw that we prepared the food for the bacteria, which we call media. So we prepared the media for them, right? And we then put some bacteria on it. So we streaked or we pour them, and then the bacteria will grow on that media, right? But you cannot do this with the viruses because the viruses, they need a living host, right, for multiplying. So if you have to grow viruses, then you need these embryonated eggs, right? So what they are doing here is that, uh, okay, again, I'm not going to discuss this technique in detail right now because that we are going to study later, okay? But I'm just showing you that you can see they have made a hole in this uh, eggshell, okay, and after this they will inject the virus in this, okay, and then they will seal this uh, uh, hole, right, and then they will put it in the incubator, and then the viruses will grow inside this egg, and then these viruses can be used for making vaccines, okay. So we will talk about this process in detail later on. Okay.
And people that have contributed to vaccines as we know them. But we've done our best to cover the larger steps as concisely as we could. And we should mention that vaccine innovation isn't a thing of the past. Work is still underway to create new and better vaccines. For now, we'd just like to stress what a modern miracle the vaccine is. The last cases of naturally occurring smallpox in the United States happened in Texas in 1949, and no cases of naturally occurring smallpox have been seen in the world since 1980. Wild poliovirus is very nearly eradicated, and many diseases that used to wreak havoc on the human race have been brought to heel by vaccination. It's truly a shame that vaccines are sometimes villainized more than the horrible diseases they protect us from. Next week, in the third episode of this series, we'll
Okay, so I think now it's clear uh, about vaccines, and yes, there is a lot more that we need to know about vaccines, which we are definitely going to read in detail. Okay, so I think I'll just stop here for attendance, and in the meantime, if you have any questions, you can ask. Okay,
Present,
so
sir.
okay, Adish, Atoiba, Ayushi,
Present, sir.
Ayushi, switch on your camera. Yes, okay. Sam, Devanshi,
Present, sir.
Ishita,
Present, sir.
Janvi, Janvi, Kshitij,
Present, sir.
Manaswani,
Present, sir.
Namrita,
Present, sir.
Nandini, Nandini, Nandini,
Present, sir.
okay. Nehal,
Present, sir.
okay. Nandini, uh, you can switch off your camera. Nehal, you can also switch off your camera. Rupal,
Present, sir.
Sahiba,
Present, sir.
Sanjana,
Present, sir.
Shakti,
Present, sir.
Shivani,
Present, sir.
Tanvi,
Present, sir.
Tridev,
Present, sir.
Vanshika, okay, Vivek, Vivek, Yukta,
Present, sir.
Yuvraj,
Present, sir.
okay. Janvi, I can see you, no issues. Vivek, Savada. Okay, so no response from Vivek. Okay, so let's continue with the class. Okay, let me share the slides. Okay, so students, this was the last slide yesterday. So we talked about the contributions of Louis Pasteur. We just saw this in the video also. So again, see, we all know that in science, a lot of discoveries occur accidentally, but the only thing is you should be able to observe it, recognize it, and then think about it. Okay. Now, in this case also, like we know about the smallpox vaccine, but here in this case, this is the first case where attenuation was discovered, right, in the lab, right? So what happened that uh, one of the person who was working in the lab of Louis Pasteur, he had to inject these chicken with the fresh culture of the cholera organism, but he forgot, and so after there, it was a very long break, right? And when they came back, they injected the same aged bacterial culture to those chickens, and the chickens did not died. They showed mild symptoms, but they did not die, and they survived, right? So Louis Pasteur observed this, and then he realized that the organism has been weakened because of its exposure to the outside world, atmospheric components, right? So then he realized that this is because the organism has been weakened and therefore it was not able to cause the disease, and that is basically preventing the these chickens to get diseased. Okay, so this was the major contribution of Louis Pasteur, and because of that, he was able to develop various vaccines. We have already seen all these things, so I'm not going to discuss this. Uh, okay, now coming to discovery of humoral and cellular immunity, right? अभी तक हम vaccines पे focus थे, but now uh, coming to other part, humans के अंदर, right? What kind of immune response is there? So there were two scientists, Emil Von Behring and uh, Kitasto, okay? So they gave the first insight into the mechanism of immunity. Okay, so they were the first one to provide us with the information about what's going on, right? What is the mechanism of immunity? So the serum from immunized diphtheria animals were able to transfer immune state to unimmunized animals. So they showed that if we take serum, okay, from immunized diphtheria animals, so uh, take some animals, immunize them with diphtheria, okay, and then you take their serum and then transfer that serum into non-immunized animals, right? Now that serum will provide protection to the unimmunized animals. So there is something in that serum. Obviously at that time they did not know what is there, but at least now they know that there is something in that serum which could provide protection to the unimmunized animals. And I think now you realize that the serum have antibodies, right? So this was the first um, time that it was explained that there is something going on in the body, something is there in the serum which can provide protection to unimmunized animal, and something happens when we immunize an animal, right? So the active components were the neutralizing toxins, which are antitoxins or basically the antibodies. There is precipitin, there is agglutination. So these are the different ways by which this happens. Okay. So another scientist, Elvin Kerbat, right? He studied the fractions of serum, right? Now this is very important. So पिछले जो 2 साइंटिस्ट थे उन्होंने बताया कि अगर हम सीरम को एक इम्यूनाइज्ड एनिमल से लेके अनइम्यूनाइज्ड एनिमल में डालेंगे, then the unimmunized animal gets some protection, right? So Elvin Kerbat, he took that serum, okay, and studied the different fractions of serum, and he observed immunoglobulin. Okay, he observed that there is a protein immunoglobulin which is responsible for all this protection thing, right? And that led to identification of antibodies. Okay, he did not give the structure of antibody, he just showed that there is a immunoglobulin proteins, okay, which is responsible for providing protection. Okay, so immunity mediated by antibodies, which is contained in the fluid or basically your serum. Okay, so this in Latin mean humor, humorous. Okay, so that's why the term humoral immunity came. Okay, so humoral basically means contained in fluid or fluidic. Okay, so antibodies in the fluid. So therefore, this term came that humoral immunity, right? So that was the contribution of Kabat, and then later on, Eli Metnik, okay, another very important contribution, and I think you know about this also. He was an embryologist studying starfish development, and then he explained that there are different cells which contribute to immune state of animals. Okay, so he observed that there are certain white blood cells which he termed phagocytes, that these white blood cells are able to ingest microbes. So that was a very big thing at that time, right? So now you know that there are some cells in our body which can actually eat or ingest the microbes, right? So he hypothesized that cells rather than serum components are the major effectors of immunity. Now just imagine, uh, again, very interesting. Before Metnikov told his observation, everyone know that there is something in the serum, right? Which Kabat said that these are immunoglobulins, right, or antibodies. So it was thought that it is the antibodies or it is the humoral immune response that is giving protection from the diseases, right? But now Metnikov, he showed that there are some white blood cells which are actually eating up the pathogen, right? So now people started thinking that no, it's not serum or it's not some protein. Yes, there may be these cells in the serum which are actually ingesting the microbes and killing it and preventing us from the diseases. Okay, so because of this observation, right, the another component of the immune response, the cell mediated immunity was developed, right? So now there were two hypotheses. One is your humoral immunity, in which the proteins or the antibodies, they are responsible for eliminating the pathogen, and then there is the cell mediated immunity, in which the cells are capturing the pathogen and eliminating it. Okay, so now there are two lines: cell mediated immunity and the humoral immunity. Okay, then in 1940s, another scientist, Marilyn Chase, uh, was able to show that immunity to tuberculosis can be generated by transferring white blood cells between the guinea pigs. Okay, so if you take these white blood cells, okay, just take the white blood cells from one animal, okay, and inject it in another animal, and then you give them the tuberculosis pathogen, and there will be some kind of immunity. Okay, so again, this experiment generated interest in the cellular immunity, right? Because we are just taking the white blood cells. Okay, so we are not taking the whole blood or serum. So we are just taking out the white blood cells and putting it in another animal, right? And that is showing protection. So this experiment, right, it again generated interest that cellular immunity is more important, right? So later on in 1950s, so by 1940s, the role of cell immunity or cell mediated immune response was like people were able to understand it. But then Bruce Glick in 1950s, he showed that there are different lymphocytes. These are the cells responsible for both cellular and humoral immunity, right? Now again, see, an interesting thing has developed. We know that there is humoral immune response, there is cellular immune response, right? And then Bruce Glick said that there are lymphocytes which can cause both humoral as well as cell mediated immune response, right? So in 1957, experiments on chicken by Glick and Zhang, so they showed that B lymphocytes, uh, so they are matured in bursa of fabricus, okay, and they are providing the humoral immunity, right? So we will be talking about the bursa of fabricus, okay. So because these cells they mature in the bursa of fabricus of the birds, right? Therefore, they were termed as B cells. Okay, and these B cells or the B lymphocytes, right? They mature in the bursa of fabricus. Okay, it's an organ in chickens or birds, and therefore they are responsible for humoral immunity. Now, some experiments were done. So, chicken जिनमें से this bursa was removed, they were not able to produce antibodies, right? Because in those animals the B cell was not maturing. Okay, so don't worry about it. So see, it's like in our body we have naive B cells. Okay, B cells which have not seen the antigen. Okay, they are just naive. Okay, and when these B cells they mature, then they encounter a pathogen, and then they start producing antibodies. Now just imagine that B cell is not maturing, right? So if it is not maturing, it will not produce antibodies. Okay, so with this experiment of Bruce Glick, right, the importance of B cell came into existence, that these B cells, they are important for production of antibody, because the birds or the chickens which were not having this bursa of fabricus where the B cell matured, they were not producing antibodies, right? So from here the importance of B cell came into existence. So later on, two other scientists, Good and Miller, okay, they showed the importance of T cells. Okay, so they showed that these T cells are maturing in thymus, okay, and they are responsible for cellular immunity. Okay, so cell mediated immune of newborn mice, those thymus were taken away, are defective. So they did this experiment. So they took the mice, okay, removed their thymus, okay, and when there was no thymus, even the T cells were not there. Okay, so it showed the importance of T cell, right? So now the role of B cells and T cells it start came into existence, right? So this is the last slide for today. So it has just uh, highlighted, right? So we have uh, talked about bearing, right? He talked about antitoxins. We have talked about Robert Koch. He showed cellular immunity to tuberculosis. right? Metnikov, he showed the role of phagocytosis, right? Uh, okay, Richet, we are not talking. He talked about anaphylaxis. So I'll talk about Charles Richet when we'll talk about hypersensitivity reactions. Then in 1919, Jules Baudet, okay, uh, he talked about the complement mediated bacteriolysis. Okay, so again, I have not talked about Baudet at this moment because when we will be reading about complement system, I will tell you the history of Jules Baudet. Okay, Carl Landsteiner, you all know, he was responsible for discovery of human blood groups. Okay, Max Thiller, development of yellow fever vaccines. Okay, Bovet, antihistamine. So I'm skipping a lot of scientists because some of these scientists you will be definitely reading when you will read those specific topics. Okay. Okay, Burnett and Medawer, they were a discovery of acquired immunological tolerance. So we will talk about this when we talk about autoimmunity. Okay, this is important. Next one, Potter and Edelman. Okay, again, what they did, right? What were the experiments that they carried out? We will talk when will we discuss antibodies, right? Because these two scientists were the one who gave the structure of antibody, right? So now we know that it's a Y-shaped structure, but it was the work of Potter and Edelman, and they also received Nobel Prize for this. So this is the list of the scientists who received Nobel Prize for immunological research, right? So there are lot many other contributions, right? Just a few slides back, I showed you the role of so many scientists, right? But not everyone gets the Nobel Prize, right? So, Yallo, he was there for the development of radioimmunoassay. This again is important. Snell, Bencraft, and Dusat. Uh, they discovered the major histocompatibility complex. Okay, so again, we will also be talking about MHC molecules. So I'm not talking about the history of that. Caesar Milstein and Kohler, they devised a method for developing monoclonal antibodies, which have a lot of applications. Okay, so we will be again talking about monoclonal antibodies when we understand what are antibodies, right? So once we understand the structure of antibodies, we will then later on study what is monoclonal antibody and what are the applications of these monoclonal antibodies. Okay. So I think I'll just stop here and I'll open it up for your questions and queries. And so I think by this time you have understood the whole story of immunology. It started with smallpox vaccine, okay, and later on, uh, Louis Pasteur, he showed that there can be attenuation, okay, and that opened up the gateway for many other vaccines, okay, and later on, scientists showed that there is something in the serum which is providing immunity, and then that something was identified as antibodies. Later on, it was shown that there are cells in our blood which is providing protection. So the two lines of immune response, the humoral and the cell-mediated immune response, it came into existence. Okay, and rest of the history I will be taking when I will be talking about the different uh, components, right? Like when I talk about complement system, I'll be taking its history. When I'll talk about antibodies, I will be talking about its history. When I'll talk about hypersensitivity, I will talk about its history. Okay, so I'll just stop here and uh, so if you have any questions, you can ask. We have 2 minutes, so if you have any questions, we can discuss.
Sir, I just wanted to send when network was not working at the time.
Vivek, you are not audible. Vivek, I cannot hear you properly. If you have a question, you can write it in the chat box. Okay, Vivek. So you were not responding at that time, so I can understand there is some network issue. Okay, so any question, any query? Okay, so maybe I can then end the class, and now we will meet on Tuesday. We have two classes on Tuesday, so we'll meet on Tuesday. And students, one more thing, start preparing your notes, okay? Because now I have to conduct uh, exam after every module, after completing every module, okay? So there are a lot of different guidelines, so I have to follow them, right? So there have to be more examinations, more uh, now more is expected from the students. So I'm sure you have heard that during your orientation that the student should come prepared in the class, and then there should be discussion on that topic, right? So I think you all should now start studying this subject. If you want to know which book, then I will suggest that you start with Cube, Immunology by Cube. Okay, so Cube is the author. So you can start studying that book. The good thing is the language is simple, so you will be able to understand. Okay. Okay, students. So I'll just end the class and see you on Tuesday.